Jablonka, qui est actuellement professeur invité du Labex Who Am I, qui travaille sur les déterminants de l'identité de la molécule à l'individu et auquel participe le CRPMS. Je suis d'autant plus content que la première rencontre avec Eva a pu se faire pour moi il y a 3 ans d'abord dans un texte qu'elle avait publié dans la revue La Recherche et qui m'avait donné envie de la rencontrer. Et alors que je suis un handicapé de la langue anglaise, mais qui comprend un peu, elle comprend le français mais ne parle pas, sur un espace de 2 ans se sont construits différents liens, renforcés entre autres par la construction de ce Labex. Et donc la voilà ici. Donc elle est professeure de biologie et d'épistémologie de la biologie au Cohn Institute for the History and Philosophy of Science et Idea à l'université de Tel Aviv. Et donc elle va aujourd'hui nous parler, elle sera discutée ensuite par Jonathan Weitzman, qui est le directeur de l'UMR Epigénétique et Destin cellulaire et un des 2 coordinateurs du Labex, par Alain Vanier, que vous connaissez tous, j'imagine, qui était directeur du laboratoire jusqu'il y a peu, et qui est donc professeur de psychopathologie à l'Univers d'études psychanalytiques, par Olivier Putois, qui est postdoctorant sur ce projet autour de l'épigénétique, sous la supervision de Jonathan Weitzman et moi-même, dans le cadre du Labex, et donc par moi-même. Alors je vais insister juste sur une chose importante, qui est ce qui se passe aujourd'hui en épigénétique a un intérêt au-delà des bouleversements et des ouvertures que ça mène dans le champ de la biologie. Puisqu'en allant vite, et Eva l'expliquera mieux, l'épigénétique a cette particularité essentielle qui est d'introduire la part et le rôle, la fonction et les effets de l'environnement sur le génome. Et donc, quelque part, ça fait sortir de cette logique où, à un moment donné, on pensait que le phénotype était complètement déterminé, contenu dans le génome. Là, tout à coup, le travail, la part, les effets de l'environnement sont extrêmement importants. Et en plus, avec le fait qu'il apparaît là quelque chose de surprenant et qui bouscule un des credos officiels de la biologie jusqu'à il n'y a pas si longtemps, qui était absolument hors de question, qui est transmission des caractères acquis par une génération à la suivante. Avec l'épigénétique, cette question retrouve une certaine actualité. Ça ne veut pas dire que ça se transmet comme ça et que c'est si simple, vous verrez la complexité de la chose. Mais d'une part, ça nous intéresse comme analyste, puisque vous savez à quel point Freud a tenu tout au long de sa vie à maintenir contre la science dominante que peut-être il était nécessaire d'envisager que quelque chose comme la transmission des caractères acquis soit indispensable pour comprendre à la fois et la vie psychique et les phénomènes de la culture. Mais l'autre point central, c'est que vous voyez bien que la biologie, en prenant en considération l'environnement et ses effets, là, ça rejoint une question de la psychanalyse. C'est comment penser un sujet en dehors de son environnement, des effets à cet environnement, de sa capacité de le modifier, d'être modifié par lui. Et donc, je crois que ce qui est très intéressant, c'est qu'avec ces recherches-là, on est devant un champ extrêmement complexe qui demande une ouverture de nos démarches, de ne pas se contenter des ressemblances de certaines analogies qui seraient des analogies faibles, mais qui peut-être dans certains cas vont se révéler des analogies fortes et qui vont ouvrir des possibilités de collaboration sans que ça signifie la disparition de la spécificité de chacun des ponts, des approches. Je dis ça parce qu'il me semble que le maître mot, et elle va le mettre en œuvre dans sa conférence, c'est la question du bridge, du pont. C'est-à-dire, avec l'épigénétique, il y a un pont qui peut être jeté entre différentes rives. Ça ne veut pas dire que les rives ne resteront pas distinctes, séparées et spécifiques, mais il y a des ponts de passage, des choses où effectivement nous pouvons voir des enrichissements de chacun des champs, des ouvertures nouvelles, et en chemin faisant, non pas de diluer nos spécificités, mais au contraire de les affirmer dans un vrai dialogue avec d'autres disciplines. Alors, je dirais là, on est à la recherche tout simplement, c'est pas sûr qu'on soit capable de le faire. Ça a été nos discussions depuis qu'elle est arrivée mercredi dernier. C'est est-ce qu'il y a moyen de trouver un objet réel, un objet de la vie, un objet concret, sur lequel on pourrait construire un programme de recherche pluridisciplinaire intégrant des sociologues, des analystes, des biologistes, des épigénéticiens. Et je dirais qu'il y a un gros challenge qui a des grandes conséquences sans doute sur les approches de la pluridisciplinarité, sur l'approche des effets de certaines découvertes biologiques qui ont des conséquences sociétales, historiques et philosophiques. Donc je dirais que c'est pour ça que c'est un vrai plaisir de l'écouter. D'abord pour l'information qu'elle va apporter nouvelle à certains d'entre vous et pour le chantier qu'elle nous permet d'explorer avec elle. Donc Eva, je te remercie et je te passe la parole.
Merci beaucoup. And thank you very much for inviting me. I'm sorry I'm speaking in a foreign language, but I hope that it will be clear. And if I speak too quickly, which I tend to when I get excited, please stop me. Right, so I'm going to, the title of my talk is The Biology of Culture, the Epigenetic Bridge. And a lot of people might sort of be surprised that there is, about a title like that. I mean, what does it mean to talk about the biology of culture? Culture is opposite to biology in many people's conception. And I'm a biologist, and for me, I see things through a lens of biology. Our thoughts, our ideas are at some level biological. And this is the point of departure, the point from which I start my thinking. So, when we're thinking about about cultural variations, and we're trying to understand different cultures, for example. For example, the Bushman culture, the Sun culture, and the culture represented on the right, which is All Souls College, Oxford. They're very, very different from each other. And yet, we do not think that the people who inhabit these two cultures have these two cultures because they're genetically different. We know that we can take a young Bushman kid, a very young child, and he can grow in England, and he can be a member of All Souls on Oxford. And we know that a child can grow in a Bushman culture and become a Bushman. So, although there are presumably genetic differences between genetic polymorphisms, as we call them, between the Bushman population and the English population, these differences are not differences that make a difference to the enculturation of these people. So there are differences, but they are irrelevant to this kind of questions. So it's, in fact, this realization that there are many, many problems that social scientists are interested in, for which genetic differences are not very important. The genetic polymorphism, the genetic differences at the level of the DNA sequence are not very interesting. It created a problem because for a long time during the 20th century, during the second half of especially the second half of the 20th century, when people were thinking about biology, they were mainly thinking about genetics, and they were thinking about genetic differences, about differences in DNA sequence. And although, as I said, there are differences, and they can be very interesting from many points of view, they were not interesting for the kind of questions that interested social scientists and many psychologists. They were simply not relevant. So once the if the biologists were trying to say to them, look, you have to think about these problems through the lens of genetics, the social scientists said, but we can't. What can you tell us? That it's all in the genes? That every problem that you have is a genetic problem? It's nonsense. We know that this is not true. We know that, for example, problems of poverty, people are not poor because they have this gene or another gene. They are poor for very, very good social political reasons. And it's nothing, it doesn't matter exactly what their genetics is. So it seems like there is no common ground. There is no way of, the discourse was not going anywhere. And there was a kind of of mutual mistrust also on both sides. When geneticists were talking to non-geneticists, they seemed to be either irrelevant or patronizing. And for the geneticists, the social scientists were talking about soft science. So the result was that there was very little communication and very little understanding between geneticists and non-geneticists when it came to problems of social sciences, to many problems of social sciences. And it came to the point, in fact, that even within a discipline such as anthropology, people from social anthropology and biological anthropology stopped talking to each other and sort of poured a lot of scorn on each other. So the bottom line is that the attempt to find DNA differences that make a difference to social life and social change and psychological problems and traumas did not work terribly well. Now, what happened during the last, I would say, 20 years is that a new kind of subject, which is related to genetics, but is not related to differences between people, differences in DNA sequence, became a very highly researched area, and this is epigenetics. I will define what, and it is a focus on how genes are expressed rather than on the DNA sequence of the gene. So two people can have exactly the same genome, exactly the same genes, but the pattern of expression of the genes in the two people are different. And the way the genes are expressed and the patterns of expression are related to development, to what happens to them to development, to what happens in their environment. The realization that this is the case, and the realization, moreover, that not only are these differences crucial for development, this is the essence of development, but that sometimes these changes in the expression of the gene can be transmitted from one generation to the next, has created a new type of discourse and has allowed people from different disciplines to understand that what they are doing is relevant to each other. What I'm trying to say is not that now we have to reduce everything to the molecular descriptions at the epigenetic level. No, we need multiple levels of description. We cannot do it. But what we do understand now is that biologists who are doing epigenetics, who are trying to understand patterns of gene expression, and people who are doing other things, people who are doing psychology, people who are interested in trauma, people who are interested in persistent social problems such as problems of poverty, have a common ground, that the level of description of the epigeneticist is although different from the level of description of the psychologist and the sociologist and the anthropologist, nevertheless, there is a common project, a common object about which they can speak. Okay.
Donc juste, François me demandait de traduire pour être sûr que... l'idée, c'est que c'est pas simplement qu'au fond, il faudrait déplacer le niveau de description de toute la réalité à un niveau épigénétique, qui serait en quelque sorte le nouveau paradigme, comme si la réalité, en quelque sorte, humaine devait être saisie à ce niveau-là, mais c'est plutôt qu'il faut se servir de la perspective qu'amène l'épigénétique pour réarticuler les différents niveaux de description. C'est-à-dire que c'est aussi méthodologique. Il y a une double... Cette avancée dans le niveau de description de la réalité permet d'adopter une nouvelle perspective méthodologique.
So, I want to define what I'm talking about. The notion of epigenetics is not very new. It was, the word was invented by Conrad Waddington, a great embryologist and geneticist, and he defined epigenetics as the branch of biology which studies the causal interactions between genes and their products which bring the phenotype into being. And soon we should see a description of it. Now, the point of Waddington, I will go to the next slide, is the point of Waddington was that what we see, we're seeing what we call in biology the phenotype, the actual appearance of the organism at the macroscopic level. Now, the phenotype is the result of development, and he depicted development as starting like up on a hill which has a lot of bifurcations, and then there are many trajectories that are leading to different cell types and eventually are leading to the construction of the mature phenotype. The phenotype, the final appearance of the mature, reproductively okay individual. Now, what is epigenetics for him? So what we're seeing at the top is what he called the epigenetic landscape. This is the developmental landscape at the macroscopic level, right? And we're seeing here a few bifurcations. Now, let's imagine that we take an X-ray through this landscape, right? So what we see is here. This landscape is actually scaffolded on interactions between genes and their products. What are the genes here? The genes here are the little pegs, black pegs that you see. There are many genes. They have chemical outputs, chemical reactions that happen because that they are responsible for. This reaction interact. They form a very big network, a very complicated network. This network, he called it the epigenotype. This is the epigenetic network. Now, this network has very interesting properties. One is that the network can be what he called highly canalized. So if we're going back here, he was talking about the fact that the regulatory architecture of this network is such that even when the things that impinge on this network there are all kinds of things that can change things within the network, but the output of the network at the macroscopic level will not change, because there are many compensations. So he talked about canalization. It is the adjustment of developmental pathways, so as to bring about a uniform developmental result in spite of genetic environmental variation. So if we think about, he was working on fruit flies, on little flies. Yes, and he looked at them. If you look at the wild type, at the normal kind of fly, they all look exactly the same. But of course they are not the same. They're genetically different. There are many genetic differences. And there are environmental differences too. Nevertheless, we don't see them, because it is like noise that is compensated by normal development. So the structure of the network is such that it compensates for a lot of variation. This is one property of every good developmental system. The other good property of a developmental system is plasticity, which is the other side of the coin. And plasticity is the ability of a single genotype to generate many different forms, many different morphologies, many different physiologies, many different phenotypes. So we have one, I'm now here, and I have particular appearance, let's say, and afterwards I'm in a different place, in a different temperature, in a different world. I'll have a different color, I'll grow fur, my phenotype will change, because there is an ability in me to change my phenotype according to all kinds of developmental and environmental signals. And this is because in the end, what is important is survival and reproduction, right? That's the bottom line of the whole story. And these two features of the developmental system are complementary and are very important. So if we go back to this picture, the Waddingtonian picture, the network has properties that, on the one hand, even if we remove one of the pegs, if we get rid of one of the genes, it will still reorganize itself in many cases, so that we will not see any difference. This is one aspect of it. And we know that, in fact, geneticists can do experiments where they knock out genes right? They get rid of a whole gene. And we don't see the difference very often. Now, the other property is that there are aspects of this network, some effects on this network, either environmental or genetic, can lead to very big changes in the phenotype, in the landscape. So we have both conservation and innovation potentialities within that are the properties of the network.
Donc très brièvement, c'est juste pour ce qui est important dans ce qu'amène Waddington, donc c'est dans cette idée de paysage épigénétique, c'est qu'au fond, alors que pour aller vite, il y a une espèce de préformationnisme un peu latent qui est enveloppé dans l'idée de programme génétique suivant lequel les gènes, grosso modo, prédiraient d'une manière plutôt fixe le développement de l'organisme. Au fond, ce qu'introduit Waddington, c'est le contraire. À partir du moment où on considère les gènes, qui sont les petites, qui sont là, d'une manière véritablement systémique, en réalité, la première propriété du système. Allô, ouais. La première propriété du système, qui est l'individu en développement, n'est plus la stabilité, mais au fond, ce qui est surprenant, c'est que globalement, il y a un phénotype des individus de type relativement similaire. Donc, au fond, on commence par introduire une idée de plasticité qui a été définie sur la slide précédente. Hein. Et au fond, ce qu'il s'agit d'expliquer, c'est le fait que, alors qu'il y a une interaction très importante entre les gènes et entre leurs produits, il y a globalement une canalisation qui permet l'existence d'individus relativement similaires. Le fait d'adopter ce point de départ, au fond, permet de mettre en avant la nature dynamique et évolutive du développement individuel.
Okay, now, like everything else in the world, definitions change, and there is a dynamic evolution of culture, including biological culture. So the term epigenetics has become very much more specialized during the last 20 years, since Waddington invented it in the 1940s. And when we're talking now about epigenetics, I'm giving you two definitions. Different definitions, because I want you to know that. But they're very similar, really. One is by Pembury, and the other one is by myself and by Marion Lamb, with whom I work. So one is the science of enduring changes in the pattern of gene activity during embryo development and beyond that do not involve alteration in the DNA sequence. These changes occur in response to conditions within the embryo and more generally. And although they are persistent, they can sometimes be reversible. Right? This is one definition. The other definition is deliberately more general. Epigenetics is the study of developmental processes that lead to persistent, self-maintaining changes in the state of organisms, the components of organisms, or the lineages of organisms. So, it is what is important about and what is the focus of the new definition of epigenetics is on the persistence of the change. Not anything that happens in development we call epigenetic when there is some kind of autocatalytic aspect to it which leads to persistence, then we're talking about epigenetic mechanisms. And we know about many such mechanisms, and I will not go here to any biochemical details because this is not important and this is not the subject of this lecture. Now, the point is, and here we are seeing, in fact, examples of plasticity, which is due to persistent epigenetic changes. What we have in all these cases are genetically identical individuals. They are exactly the same genetically, and yet, phenotypic level and epigenetically, they are different. So we have two identical twins here and there. One identical twin up there is a normal little girl, the other one has a problem, has a mental problem and physiological problem. There are two identical twins here. There are also differences in them, for example, differences in sexual orientation. They're exactly the same genetically. They came from the same fertilized egg. We have two mice up there. They are not identical twins, but they come from the same, what we call isogenic line. That is line where there was many generations of brother-sister breeding, matings. As a result of that, the individuals are similar genetically, but as you see, one is fat yellow mouse, and the other one is a brown normal-looking mouse. The fat yellow mouse also has diabetes and all kinds of prone to cancer, all kinds of problems. The difference between them, in this case, we know is environmental and is due to the diet of the mother. The diet of the mother of the brown mouse was rich in folate. The other one didn't have folate. And this is the difference between, and this is responsible for the difference in the big difference, which is epigenetic difference. In this case, we know exactly where the epigenetic difference is located. The actual sequence, which in one case has one type of expression and one type of structure. One type of structure, not DNA sequence. The DNA sequence is the same, but something imposed on the DNA sequence is not the same, is different. And here we have two insects. They are the same, but genetically, but they grew on, there were parasites, it's a parasite, there were parasites on a different type of plants. And the result of the plant on which they grew, they became very, very different from each other. And you can think about many, many other examples. For example, in a colony of bees or a colony of ants, we have the queen and we have the workers. They are enormously different from each other, but the difference is not due to differences in their genes, it is due to the diet that they had as very young larvae. And these changes are persistent, and we know, for example, in the bees, that there are epigenetic changes that are underlying this. Okay. So, just a very, very simple picture without going into any biochemistry. If we're looking here at this 6 bits of DNA, the 6 bits of DNA are exactly the same in terms of sequence, but the different colors tell us that they have different and persistent expression patterns. Okay? This is all you have to know. And just to know one other thing, because I will say a little bit about it. One of the epigenetic aspects that we understand better than others is something we call DNA methylation. So DNA methylation is something that is imposed on the DNA and can lead to changes in gene activity. This is an epigenetic marker.
Donc la slide précédente, l'idée c'était simplement de montrer que les changements, viennent de l'environnement uniquement. Les exemples ont été choisis pour ça. Ça peut être l'alimentation de la mer, etc. C'est plein de choses. Et le deuxième point, c'est qu'ils sont persistants. Voilà. Cette image-là, c'est juste pour introduire le terme de méthylation qui va revenir parfois, qui est c'est l'un des effets épigénétiques possibles, l'une des traces ou marques épigénétiques possibles sur la base, encore une fois, d'une séquence d'ADN identique.
Okay, I just want to give you one example because you're dealing with this kind of, I guess, many of you are dealing with problems related to this. Now we all know that there are people who are suffering from autism. And here we have a recent study from 2013, where people looked at identical twins that were different, that one of the identical twins was normal, the other one was autistic. And we know that there is a whole spectrum of autistic spectrum disorder. Some are very, very badly autistic, and some have Asperger, and so on and so forth. So we had these two twins. So we had a group of twins, and in 30% of identical twins that have autism, we have discordance. So that one twin is normal and the other one has autism. Now, so the genetics of these twins is the same. What is the difference between them? So now that we know how to look at epigenetics, at these patterns of persistent gene expression, and the thing that leads to persistent gene expression, for example, DNA methylation, what they did was they took blood. We can take blood. We don't have to go to the brain. We can take blood, general blood, and we can look at the blood, and we can see whether the methylation pattern is the same or not, and the blood is informative, fortunately. And what they saw was that not only was there a difference between autistic and the autistic twin and the non-autistic twin, and it was a big difference, statistically significant. It was not just noise, because we have a lot of variation, just variation between any individual. But also, the difference was growing with the severity of the difference between the normal twin and the autistic twin. So it seemed like there is something causal here. Now, again, in this case, we don't know what was the environmental, if there was a specific environmental reason for the difference, whether it was some kind of developmental noise, we don't know. And we should not jump to conclusions. But we do know that this is the type of difference we see between these twins. Okay. Now, when we think about a human being, which is what we're interested in here, and we think what makes different human beings what they are, what is the input into the development and into the heredity of a human being, what are the differences that make a difference, we have to think about many, many things that go into the construction of development. So first of all, as a geneticist, of course, I can tell you, genetic differences can make a difference. They don't always make a difference, but sometimes they do make a difference, and sometimes a big difference. Another thing that can make a difference are epigenetic differences that appear already at the stage of the egg, and already at the stage of the sperm. And also, and they appear at the non-DNA parts, so it's not a genetic difference, it's an epigenetic. There are also differences that are due to the kind of womb that the individual is developing in, and the early care that the individual is receiving. These things can have very long-term, persistent effects on development. And we know, as human beings, we're living in a symbolical world, and therefore we know that the kind of social world and the kind of culture in which we live can have very profound and deep effects on our development as human beings, as social creatures. And the niche, the ecological social niche in which we are. So this is a picture just to give you a kind of, it's called the Five Mothers. And I apologize for there being no fathers, it's just for simplicity. It's, but there are fathers. Of course, the father is very important. But what we have here are the five mothers, and these are the inputs. And this is, in a way, something very trivial. Everybody knows that there is a genetic input, there is this epigenetic input into development, that early care, that what happens in the womb is very, very important, what happens in early development is important, and the big mother is mother culture, right? All over the place. So you have the DNA mother, the epigenetic cellular mother, the uterus mother, the early development, early care mother, and the great, great mother culture, which embeds everything. Now, all this we knew. There's nothing new about it. What is new about it is that we now realize that all these inputs are important not only for development, but they can also be important for heredity. So it's not just we thought that the only thing that is really important for heredity is in the DNA. All the rest is important for development, but not for heredity, and therefore also not for evolution. But now we realize that this epigenetic marks can be important for heredity too. They can be passed from mother to offspring, from father to offspring. And we know that what happens in the womb can affect very greatly the fate of the child and more. And it can carry over even to the grandchildren. And we know, of course, that symbolic, that there is a lot of symbolic transmission in our species. There is also behavioral transmission, not symbolic, in other species of animals, which is also important. We have it too, but and so on and so forth. So we understand that there are many inputs, not only to development but also to heredity.
Oui, donc ce dessin, elle s'excuse du fait qu'il n'y ait pas de père. Elle dit bien sûr les pères sont importants, mais bon, pour des raisons de simplicité, les mères sont ici. Donc c'est les 5 mères qui correspondent aux différents paramètres, on ne va pas revenir dessus, mais qui déterminent l'évolution de l'individu. Et encore une fois, là, elle réinsistait sur la dimension de passerelle, au sens où ce que par ailleurs l'épigénétique remet en avant, c'est le fait qu'on ne peut pas véritablement séparer le niveau du développement de l'individu du niveau de l'hérédité, au sens où ce qui contribue au développement de l'individu, de fait, peut être, dans plusieurs cas sur lesquels on va revenir, passé aux générations suivantes. C'est la réarticulation entre l'ontogénèse et la phylogénèse que l'épigénétique permet de repenser.
Yes. So we have the definition, therefore, of epigenetic inheritance. And epigenetic inheritance and epigenetics is not exactly the same term, although it gets confused in the literature. Epigenetic inheritance is a component of epigenetics. It occurs when phenotypic variations that do not stem from variations in DNA base sequence are transmitted to subsequent generations of cells and organisms. It's a very simple, straightforward definition, and that's the definition we're using. Now, what I want to talk about now is, the title of my talk was The Biology of Culture. So now I want to see how culture changes biology and vice versa. So, first of all, there are changes. What happens to us, even what happens to me now, for example, when I'm talking to you, that changes in my brain. And when I live in a particular culture, my brain is reflecting this culture in many, many ways. The patterns of activity that I have in my brain are different from the patterns of activity that I would have in my brain had I lived in a different culture, for example. There are all kinds, the culture, the social environment in which I live, the culture in which I live can change the pattern of expression of my genes, my epigenotype, my epigenetic marks, and this can lead also to transmission to the next generation. And the social and cultural innovations that can be introduced to a society can alter not only the epigenetic and the brain circuits, but they can also change genetics, in that they construct the environment, a particular environment in which genes are selected. And I will give you examples for all these things now. Now, I want to start with a nice quote that I like from an anthropologist, Smith and Parson. They say, by changing the cultural environment, each generation changes the brain of the next. Much of this occurs unintentionally, hence our description of the brain as an artifact of culture. However, when we deliberately seek to influence the developmental trajectory, such as by provision of particular educational environments or dietary supplements, we are in fact turning the brain into a cultural artifact. And I like this because I think it is both common sense and surprising, because we don't usually think in this kind of way. And I just want to give you a very simple and very well understood, relatively, example, and this example is literacy. Literacy has been invented several times about 5,000 years ago, but for a very long time it was only very few people could read and write. It was only the elites that could read and write. But during the last 300 years, most people in many countries can read and write, so it's very common. Now, the one thing, when a child, and not only a child, even an adult, a normal adult that for all kinds of reasons has not been exposed to learning to read and write, is learning to read and write, there are changes, and specific changes, in brain circuitry. In a bit of the cortex, which is called the occipital lobe on the left-hand side. And so there is a reorganization of the brain patterns of the brain in this region. And not only this, there are also morphological changes as a result, not only in children, also in adults, normal adults that learn to read and write. For example, the corpus callosum that connects the two hemispheres gets thicker, and there are other changes in the amount of grey matter in particular regions. So there are actual morphological long-term changes in the anatomy and physiology of the brain as a result of a cultural change. Thus, the genetics is not very important here, although it is not totally unimportant, because there are people that are dyslexic because of particular, among other reasons, they can be dyslexic because of certain genetic variations. We were not selected genetically to read and write. That's why there is a very big variation in the population with respect to this trait.
Juste une pour pousser le petit exemple sur le littérisme. Il y a des travaux notamment de Stan Dehaene, mais pas seulement lui, qui ont montré donc non seulement qu'il y avait bien sûr une neurogenèse et une réorganisation du lobe occipital, une partie du cerveau suite à la lecture, mais aussi que la structure morphologique d'ensemble du cerveau évolue en raison de l'exposition répétée à ce type de tâches. Donc c'est un changement qui s'inscrit dans le long terme et qui ensuite est stabilisé et canalisé.
Now, the next aspect I want to talk about, I said that there are epigenetic differences that are the result of cultural changes. Now, unfortunately, war is something that is characterizing our species, and there was a great famine in the German-occupied part of the Netherlands, in Holland, during the winter of 1944-1945, near the end of the Second World War. So there was a German blockade that blocked food, cut off food and fuel shipments from the farms to the Dutch because of they didn't help the Germans in their war effort. And about 4.5 million people were affected by this famine. And about 22,000 died because of it. Now, the Dutch afterwards, those that survived, most survived. And the Dutch have very good health records. So one can follow what happened to the kids that were born during this time. So the research on the children that were conceived during this period, whose mother were pregnant during this period, especially the second trimester. It depends on the trimester exactly what type of problem they have. For example, in the second trimester of another pregnancy, they found that there is an increased incidence of schizophrenia in these children, statistically significant, and also schizotypal personality and various neurological defects are more common. And this is very often being shown many years afterwards, during adolescence or even later. But we can see that there is a significant change. The other thing that people did was to look at the epigenetic marks in these children compared to their brothers, for example, who were born from the same mothers and fathers later or earlier before the famine, to see whether there are any kind of consistent changes in the epigenetic marks, and there are. So now they're looking into the third generation. Fortunately, it goes down, but there are still some traces. So, we know, so this is an example that shows us that a political, social, cultural change can affect the epigenetic profile of a population. Right. I want to describe, so what do we do with this? How do we, there are obviously many, many inputs into human beings, into their culture, into what happens to them. How do we think about culture? How do we think about a human being that is embedded in a culture? And what I want to suggest is a kind of theoretical perspective on this question, which is influenced and is based on developmental system theory. So, if we're looking at things from a from a developmental, evolutionary developmental system theory, kind of evo-devo, as we call it. A cultural system is a dynamic entity into which individuals are introduced, in which they develop, and to which they contribute. This system or network gets reconstructed, of course always with modifications, using inputs mediated by past and present individual and collective activities and products. I think this definition is more or less acceptable, not only by sociologists, for example, and anthropologists. They would agree that this is a relatively acceptable definition of culture. Now, if we're taking this approach, what we want to understand is the dynamics of the development of this developmental system. We want to follow how it changes or how it persists over time. So, we saw the picture of Wellington, and I want to use this picture as a model for thinking about culture. Okay, so we have here we see here 4 developmental trajectories that are leading to particular points of stability. These points we should call attractors. It's like points where the system tends to reach and evolve towards and stay there, even if it starts from slightly different points. So, okay, so remember this picture. Now, here we have another picture. Just imagine society, the world, the social world, as a kind of landscape, where we have different societies and they're separated by, like, their valleys. This is where the attractors are, and people are sort of being led to the attractors. They develop along different trajectories and reach attractors. Of course, it is not a completely deterministic system, because people can move between societies, but it's not very easy to move between societies. We know this. People tend to stay if they can. They are not forced. I mean, you can push a person. We can see here some pushing of the boy, which is an individual, between different attractors, between different valleys, between different social systems. But usually, people tend to go to the attractor through paths that were partially constructed towards a particular typical social attractor in which they were born. So, I want to talk about something that we call, now, what I'm talking about now, and then when I'm saying we, the we is myself and my colleagues, Ido Tavori and Simona Ginsburg. And so it's not a royal we, just the people who did the work. So the social landscape is a dynamic pattern of life in a particular community. It is the outcome of ecological, social, and epistemological niche construction. People construct the world in which they live. And an individual develops within and contributes to the collective and cumulative interaction that builds up the social landscape. Now, what we have here is a kind of cut, transverse cut through a social landscape, and the little balls are the people. So the trajectories that lead to the attractor in the social landscape are the developmental paths of individuals as they become socialized. Individuals can affect their own and other individuals' trajectory to some extent. Individuals go through partially pre-existing trajectories, because we're not born into a tabula rasa, we're born into a social world which is structured. And we tend to follow the trajectories that already exist in this world. And people can also stabilize, deepen, and sometimes deflect the trajectories that exist in this landscape through their activities. There is a very famous Chinese saying, when many men walk the same way, a road is made. So when many people go through a trajectory, a road is made, and it becomes easier to follow it and so on. And the social attractor is that region in the landscape where individuals settle. That is, they are identified by the researcher, the sociologist, the anthropologist, the psychologist, as relatively typical members of the social setup, and they typically also contribute to the maintenance and the stability of this setup. And always the particular social landscape on which we're looking at, we have to focus on something, is part of a larger social landscape, but interactions within the particular landscape in which we're interested are much more numerous than interaction between landscapes. Okay, it's a kind of hub, so to speak. Now, this is very, very general and very abstract, I know. So I want to give examples of this kind of phenomenon. And I will give 5 examples, and I will show you 5 different inputs. And then I will finish with an example of how we can study a system like that. So the first example that I will give is urban poverty in the USA, where we're looking at social, symbolic, and epigenetic interactions. The second one is Orthodox Jewish community persistence in Los Angeles, where we're looking particularly at social symbolic interactions. And the third one is connexing deafness in the United States of America, deaf-mute people, where genetic and cultural interactions are very important. So we're not neglecting the genetic. It is relevant sometimes, but not always. Okay, here we have urban poverty in the USA. And what I'm giving you is just a sketch. It's very little about this case. So in the USA, children are born into the lowest quintile, 20% of the social economic ladder, have only 1% of ending at the top, 5%. And this is even much more exaggerated when they are being born to much lower social strata. 63% of black children born into the lower income quarter will remain there as adults. And their children will also be poor. And this percentage is even higher if we're going even lower. So what is going on there? Why do we have this persistence of poverty? So there are many social cultural factors, and they include the kinds of jobs available, the education of parents, the quality of school in the inner city, the structure of the state welfare system. We have meaning-making processes that people use in order to make sense of their life and to be able to go on living, but in a strange kind and tragic kind of way, these processes are themselves reinforcing their acceptance of their fate. So, this, for example, agreeing to work in horrible back jobs, which are underpaid because they want to feel respectable, and they'll never get enough money to be able to educate their kids or do anything, get out of this, or becoming part and being proud of becoming part of a drug culture and things like that. There are also biological and epigenetic factors. For example, cheap food, which is high in low in good nutrients and vitamins, and very high in fat and sugar, is not good for you. And it leads to obesity. People in very poor areas also very often drink alcohol. There is a lot of alcohol syndrome there. They take drugs. All these things have effects on the offspring, straightforward effects of the offspring. The offspring are not born with the same initial in order to overcome what they have gone, you have to input a lot. They are not born on equal terms with bourgeois children, right? So there is a lot of that too. And people who do make it, who do manage to get out of this system, they don't contribute to the system. They cut, usually, typically, they cut any connection that they have to the system. So this is again, they do not help, and they form a kind of gap between themselves and the other. All these things create a network of interactions which is self-sustaining. So it's very easy to see why, if you are starting poor, you may well end up being poor, not only your children and grandchildren. Of course, people do get out of there. So it is not totally determined, but it's not easy because of these factors. Right, so this is one example, and here we saw epigenetic and other factors. Now, I want to give another example, and this is an Orthodox Jewish community in Los Angeles, in the Beverly La Brea neighborhood, an area that is well known for its secular and transgressive youth culture. So in this, the Jewish people are living in this area. How do they manage to conserve their culture? And not only to conserve it, it's a thriving community, it's growing. How? What are they doing? How does it persist? And we're talking about several generations already. So, you know, they are living in this culture and that's what they do. Right, so what's happening there in the term that we're talking about? So Eva Jablonka did a very big work on this community, on the factors that are preserving and constructing the everyday life of this community and that are leading to its persistence and to its thriving. So I'll just give you a few examples. So they have institutional frameworks that generate a lot of participation. For example, in schools, in the synagogue, there is a lot of participation of adults in many, many activities there. And that creates multiple obligations that the members are all the time pulled towards. So there is a very strong gravitational power towards being part of the society and taking part in its activities. Now the other thing is that because they are similar to others in many, many ways, they are making boundaries between themselves and the others and transforming the marginal difference between them and them into a very seductive kind of experience of community that is different. So there is a very, very strong attempt to celebrate the difference. And there are many public worship situations that create interactions that suspend the rigid communal life and generate communal meanings. Many, many, many different types of these things. And together these things create a community which is very cohesive and very well differentiated. The boundaries are quite clear between it and its neighbors. And these are just a few examples. There are many parameters like that that are creating a very complex and very interesting network that is making this community into a very distinct entity. So here, but in this example we had mainly social, kind of social and symbolic kind of aspect that are recruited into the construction of the society. I don't think that there are many genetic aspects, and I don't think that there are many epigenetic aspects, but maybe they are. I don't know about them. I don't think they are very important. Now I want to give you an example where the genetic aspect is important. It's a very interesting example. The observation is that the frequency of connexin deafness, which is which is the most common deafness, genetic deafness, has increased, has doubled in the last 200 years. And the question is why? Why are there more deaf people in the United States than there were 200 years ago? And the answer is that this is happening because of the invention of sign language. Sign language was introduced at the beginning of the 19th century, and it altered the reproductive pattern of deaf people. It did two things. The first thing, which is less significant in some way, is that deaf people got more children on the average than they did before. So the frequency of the gene for deafness increased slightly, but not very much. It did increase a little bit, because they were more accepted into society, because they were considered as normal people, because they could function cognitively a lot better, because they could communicate through language and so on. But the main reason was that deaf people tended to marry other deaf people or relative of deaf people. And the reason that they married deaf people or relative of deaf people is simply because they couldn't communicate with them through sign language. These are the people who know sign language. And therefore, the frequency of homozygotes, this gene is recessive. So you need 2 copies of it for a person to be deaf. And the probability of somebody who is homozygote and is deaf marrying somebody else who either is deaf and also is homozygote or is carrier of a gene, he can speak, but his brother, for example, or one of his parents is deaf, and therefore he knows sign language, but he can speak, but his carrier is this leads to the increase of the homozygotes in the society. So what we have is this situation. So if we are looking here at the top, at the increase of frequency of the connexin gene, we see that it goes slightly up, but not much. But if we are looking at the top graph, we see that because of this non-random marriage, non-random mating, it's called in biology assortative mating, positive assortative mating, you mate with those who are like you in phenotype. As a result of this, there is an increase in the frequency of homozygotes. So in this case, what we have is a very interesting interaction between genetics, between societal change that is changing the environment and changing the pattern of life of human beings. It's changing also slightly the selection of the gene, not very much, but a little bit. It's making this gene more fit, so to speak. But more importantly in this case, it is changing the nature of the matings and therefore the number of homozygotes in the population. So here we have an interaction between, here the culture is creating the conditions for altering the genetic constitution of the population. So, now what these examples tell us is that if we're thinking in these terms, if we're trying to understand which are the components that are important for the creation, for the description of the social system in which we are interested, we can identify the relevant factors and the processes that make the particular social landscape in which we are interested without a priori privileging one particular factor. Sometimes genetics is important, sometimes it is not. Genetic differences are not important. Sometimes epigenetics is very important, sometimes it isn't. Symbolic culture is always important for human beings, by the way, so we can never, ever get rid of it. So it's always there. But what this way of thinking gives us, it facilitates the consideration of stability. It helps us to understand why there is stability, in spite of the many changes that all the time happen in the world. And the world is changing, it's dynamic. And it also explains to us, if we're thinking about these networks and the way that things interact and the autocatalytic kind of cybernetic regulatory interactions between things, it helps us to understand why sometimes we can have very sudden jumps in the system. Because this is exactly what is happening in complex systems. And we have now models that allow, from complexity theory, that are allowing us to understand how this type of systems behave. And the other thing is that when we're thinking in this way, we are no longer making this distinction between evolution and development. They are continuous processes, because developmental changes are because they are repeated from one generation to the next, and because they scaffold what happens in the next generation, they have direct evolutionary consequences. And so evolution and development are continuous and are dominated by network dynamics. And we need to consider different timescales. When we think about it, we need to consider the timescale of the development of the individual and the socialization of the individual as it develops as a member of a particular community, and the timescale of the community itself and the life cycle of the community. So at least these two timescales we have to consider. Now, there are many things that we can ask about this. If we are looking at stability, we can ask about dynamic stability. We can ask what is the strength of canalization for different cultural phenomena. For example, different poor environments in different societies. Will they have the same structure? Will they have different structures? Will one structure be more easily changed? Can it be more easily changed or not? How robust it is and things like that. How early events can scaffold later ones. And if we analyze this kind of patterns, maybe we can see which interactions are particularly important for the construction of the particular pattern that we're looking at. For example, which factors and which processes participate in positive feedback loops that sort of feed the system again and again and lead to the repetition of cycles, all kinds of cycles, good and bad. And but of course, social systems change, so how do they change? So they can change in many, many different ways. No? Okay, never mind. So they can change in many different ways. We can, for example, there can be changes to two landscapes can come together. For example, the Jewish Enlightenment, which is called Haskalah, in the 18th, 19th century, it was a movement that was advocating integration within general non-Jewish culture. And this was the result of political and social changes in the non-Jewish society. And two landscapes that before that were very separate came partially together. And that created massive changes within traditional the Jewish landscape, and created a new hybrid landscape between these two landscapes. For example, we can think about all kinds of internal conflict that for a long time are cryptic, don't have any effects, and then at a particular point they can drastically change a landscape. We can think about communication technologies and the way that they change social landscape by bringing information, ideas, migration of ideas and information between different societies. And we can think about all kinds of cascading effect. If we start from a particular initial condition, there will be a kind of dynamics, cascading dynamics, that can lead to a change in the society that will breed another change and another change in a semi-predictable way. And I want to give an example of an interdisciplinary project that one can do from this kind of point of view, that brings together both epigenetic, psychoanalytic, psychological, social, sociological, anthropological aspect. First of all, I want to say that if we want to do an interdisciplinary research, every real life problem is by definition interdisciplinary. That's the nature of the beast. So, okay, so I will give you an example, and of course, every example that I give, and also, as you can imagine, this lecture too, is driven by my agenda, and my agenda is, I'm a socialist, and I'm interested in trauma, and I'm interested in the construction of identity, of collective and individual identity. So this is the point of view. This is why I'm interested in these things, and I'm just telling you so that you know where you are. Now, if we want to look, I'm just giving you just a hypothetical example of how we can study a particular social network, a social landscape, from a developmental system point of view. So let's think about a community that arrived to France in 1994. They're from Rwanda. Right? There was a huge, terrible genocide in Rwanda, and people arrived, and the people who arrived were very, very stressed. I'm talking about the Hutu people. You know what happened. So we're interested in this community, let's say, and we want to understand whether the trauma that happened to this community, how did it affect it? How did it affect the people? How did it affect the well-being of the people, their sense of identity, their sense of belonging to the French culture or to the Rwandan culture? Who are they? Their sense of who they are. So we need a lot of data in order to answer this question. And we're going to, as scientists, we have to do it very, very systematically. So, and look at a lot of things. So for example, we want to know about the mother, when the mother was exposed to stress, what kind of stress it was, what extent of stress it was, whether she was exposed before she got pregnant, during pregnancy, after birth, during several periods. We want to know whether she had postnatal depression, whether she could look after the child, what was going on with her in her functioning as a mother towards the child. But we also need to know about all the things that happened in utero when she was pregnant and even before she was pregnant, because we already have data for rats and for mice that things that happened before pregnancy can affect the hormonal profile of the animal and can affect the development of the fetus, even before pregnancy. Okay, we also are very interested in the father, because the sperm is not just carrier of DNA, it's carrier also of epigenetic marks, among them a lot of little small RNAs that are regulatory molecules that are very, very important. It can alter the fate of the child, so males also have to be responsible for the next generation. So we want to know when the father was exposed to stress, period before impregnation, its function as a parent during early and later childhood. Was there specific problems? Was it abuse? Was he depressed? All these things we need to know. We need to know about both parents, how they interact within their society, among their community, their socioeconomic status, their health status, their access to education, their self-identification. All these things are crucial. We have to know about the offspring, the children. We have to know when they were exposed to stress in utero, postnatal, early childhood, a combination of these things, for how long, how severe it was. We need to know, we have to divide the data into different groups. For example, was the child conceived in Rwanda to traumatized parents but born and grew up in Paris? Was it conceived and born in Rwanda and immigrated as an infant? Was it conceived, born, and experienced trauma for several years and then immigrated? What's going on? So we have different groups, and so we also have ability to see the extent of how these differences are related to the well-being of this individual. Now, the kind of information that we want is about mental and physical effects on the individual, about the individual's perceived identity. We need data from doctors, psychologists, psychoanalysts, teachers, social workers, anthropologists, and probably a lot of this data is available from the health and social services and from the statistics community. We need epigenetic data. We know now that we can look at epigenetics of target genes, genes that are related to mental problems, like in the autistic kids, but not only, there's also related to schizophrenia and other things. We can look at blood and see if there are differences in the epigenetics of children belonging to the different groups. So we need data from molecular biologists, from epigeneticists. And since it's already 20 years, and some of the kids were young when they arrived, but they can already be parents, we can look at the transmission to the next generation also. And once we have this information, we can construct a model, a good model of the social landscape, and we can begin to understand what are the interactions between the different factors, where are the best places to intervene. We can't know what are the best places to intervene if we don't understand the interactions between the different factors in this particular landscape. So this is just an example. What I'm trying to say is that we can, if we're thinking about a real life phenomenon, social phenomenon, we have to take into consideration really all these aspects. It will include also the epigenetic aspect, but it is just one factor that is an input into the system among many. It is a factor that is allowing us, fortunately, to have a productive dialogue and a real discourse between biologists, psychologists, and social scientists. Thank you very much.
Merci beaucoup, Eva. Moi, je ne vais pas engager la discussion tout de suite avec elle, parce que ça fait plusieurs jours qu'on discute ensemble et ça va continuer demain, et donc je trouve que c'est intéressant aussi qu'il y ait d'autres aspects de discussion. Je ferai juste deux remarques. C'est que finalement, le Labex, le laboratoire, sont des paysages sociaux qui doivent fonctionner comme des attracteurs de recherche, et ça serait intéressant si on arrivait à faire ça, et à infléchir les trajectoires trop établies des parcours académiques soumis à évaluation et compagnie. La deuxième chose, c'est, vous voyez toutes les pistes que ça ouvre, mais un des problèmes tout de même, c'est comment dans une situation comme ça, être d'une extrême prudence, parce qu'il ne s'agit pas de conclure trop vite, qu'il ne s'agit pas de faire des ponts trop vite, qu'il ne s'agit pas trop vite, par exemple, moi je pourrais dire, génial, ça démontre que Freud avait raison sur la transmission des caractères acquis dans Moïse et le monothéisme. Il ne faut pas aller aussi vite que ça, donc il faut à la fois avoir de la prudence méthodologique, mais vous voyez bien que l'objet que propose Eva à la fin, c'est tout de même un champ d'innovation, d'invention et d'audace qu'il faut être capable de mener. Alors, quelque chose, je crois vraiment, le point où on en est aujourd'hui d'un certain nombre de possibilités de collaboration, c'est à la fois l'extrême prudence méthodologique et épistémologique, mais ne pas avoir peur de prendre des risques. Et c'est ne serait-ce que pour ça, je pense, en dehors de toutes les informations nouvelles qu'elle apporte pour certains d'entre vous, je tiens vraiment à te remercier, Eva, de cette remarquable conférence. Alors, qui Olivier, Jonathan, Alain, ou quelqu'un de la salle? Tiens, Alain. Viens. Allez, venez là. Alors, Jonathan Weitzman est professeur de biologie ici, et donc, comme je le disais, la responsable du laboratoire d'épigénétique et destin cellulaire de notre université.
Bonsoir. Do I speak in English or in French? תודה רבה, אווה. מאוד נהניתי מההרצאה. Je voulais juste remercier Eva pour son arrivée. Et, donc, je suis biologiste. On n'a pas vraiment l'habitude de commenter et de discuter des présentations, mais j'ai quelques remarques à faire. Et, donc, comme elle a dit, Eva, l'épigénétique, pour une raison qui n'est pas très claire encore pour moi, attire des réflexions très très personnelles. Et donc, je vais ajouter, sur ce qu'elle a dit, Eva, quelques réflexions personnelles, focalisées sur le mot pont, le bridge. Et la première chose, c'est de remercier Eva, parce qu'Eva est quelqu'un vraiment qui vient de la biologie, qui a travaillé sur la méthylation avant que c'était à la mode, et qui a fait toute sa carrière, une carrière de pont. Entre les disciplines, qui est un peu un exemple pour nous, et on a vu dans les quelques jours qu'elle est là, à quel point c'est difficile de jouer ce rôle de pont entre les disciplines, et c'est un peu un exemple pour nous. Nous travaillons tous dans une université qui est censée être interdisciplinaire ou multidisciplinaire, et ces dernières années, grâce au financement de Labex, on a commencé à travailler cette idée de pont, de bridge entre les disciplines, qui n'est pas toujours facile, mais qui est un peu pour ce siècle, pour le XXIe siècle. Et quelque part, l'épigénétique est le pont entre le XXe et XXIe siècle. Et pour mettre ça en évidence, j'aimerais juste toucher quelques éléments qu'Eva a peut-être moins dit. C'est qu'en 2001, on a publié, on a complété le séquençage du génome humain. Et donc on a rentré dans le XXIe siècle avec le génotype. Et vous serez tous, votre ADN peut être séquencé demain, c'est sur l'ADN de vos enfants, donc le génotype de vos enfants serait disponible. Et donc la question de l'interaction entre l'environnement et le génome, ou la génétique, est une vraie question, parce d'un côté, on a déjà le génotypage, le génome, on va avoir dans notre main, qui laisse l'autre côté du discours, qui est l'environnement. Moi, je dis souvent à mes étudiants que quand un chercheur biologiste parle de l'environnement, il utilise ça pour dire « je ne comprends rien ». Et donc, les choses que je connais bien, c'est le génome, et tout ce que j'arrive pas à expliquer, c'est les impacts environnementaux. Et c'est pas satisfaisant pour un chercheur de parler de l'environnement comme quelque chose qui est mal défini. Ce qui est intéressant dans l'épigénétique, et c'est pour ça qu'elle fait le pont, c'est que, en plus des idées conceptuelles qu'Eva a parlé, aujourd'hui, on a des outils moléculaires et mécanistiques pour arriver à vraiment comprendre quel est l'impact de l'environnement sur le génome. Et donc, c'est les marques qu'elle a mentionnées. Je ne vais pas rentrer dans les détails techniques. La méthylation de l'ADN, les modifications des histones, l'acétylation, elle a parlé un petit peu des petits ARN. Donc, il y a plein de... Et donc, si ça vous intéresse, dans mon laboratoire, on étudie beaucoup ces aspects très moléculaires. Mais on a aujourd'hui des outils très sophistiqués pour mapper, pour regarder au détail, lettre par lettre dans le génome, où se trouvent ces marques qui sont l'impact de l'environnement sur le génome. Et ce qui est intéressant dans l'épigénétique, c'est l'épigénétique, la raison pour laquelle c'est séduisant, c'est que ça nous arrive ça nous permet d'introduire l'idée du temps et du mémoire du temps avec la génétique, qui était quelque chose qui était... La génétique est un peu statique, elle n'a pas un dynamisme que quand la Wellington a introduit. Donc, la cellule souche ou l'embryon traverse cette montagne et l'épigénétique nous permet d'intégrer avec la génétique classique l'environnement et le temps, donc les traces de l'environnement sur le génome. Et aujourd'hui, si vous êtes intéressé, on peut discuter pendant des heures des techniques. On a des techniques très sophistiquées pour regarder ça. Et donc, ce n'est pas juste une promesse qu'on peut parler un peu vaguement de l'impact de l'environnement, on peut vraiment le tracer. Et donc, tous ces exemples qu'elle a parlé de Dutch Winter, Hunger ou l'impact de l'interaction entre mère et enfant, et on peut vraiment arriver à tracer ça et commencer à décider, à comprendre ce pont entre environnement et génome. Did you understand any of that? Yes. Au niveau très scientifique, très technique, l'épigénétique est aussi un pont entre, donc elle n'a pas vraiment dit, mais elle a touché sur ça, entre l'état cellulaire, donc le phénotype quelque part, et le métabolisme de la cellule. Donc on parle que la nutrition a un impact sur le phénotype, qui reste un peu conceptuel. Aujourd'hui, on peut disséquer ça au niveau moléculaire, de voir quelle partie de la nutrition a un impact sur quelle partie du réseau métabolique et un impact sur l'état cellulaire et les phénotypes via ces modifications épigénétiques. L'autre chose, juste le dernier point elle a beaucoup insisté, mais qu'on a parlé même ce matin, c'est l'année dernière, Takahashi a eu le prix Nobel pour une découverte sur la plasticité cellulaire. Quelque chose qui était imprévisible, qu'on peut prendre une cellule somatique et par quelques manipulations expérimentales, créer une cellule souche qui a la pluripotence. Donc on peut prendre une cellule qui est tout en bas de la montagne et le pousser tout en haut et après l'aider de prendre de notre chemin. Pour un biologiste, c'est incroyable que le système cellulaire a cette plasticité et tout ça sans changement en principe de génome, donc tout ça basé sur un changement de l'état épigénétique à n'importe quel moment de la montagne. Donc, intellectuellement, c'est incroyable. Pour la santé, c'est un énorme impact parce qu'on peut prendre une cellule somatique d'un malade, créer une cellule souche dans une boîte de pétri et réduire les cellules qu'on a besoin pour une thérapie cellulaire. Donc on peut régénérer aujourd'hui n'importe quel type cellulaire dans une boîte de pétri. Mais j'essaie de convaincre Eva ce matin que l'idée de plasticité, offre, je crois, un optimisme que les phénotypes et l'état cellulaire, mais aussi peut-être les états sociaux, sont plastiques. Donc si on peut arriver à comprendre comment changer l'identité, recréer la potentielle et redériver un autre chemin. Donc je crois que ça offre pour les sciences sociales et humaines un optimisme que la vie, cette plasticité de la vie nous permet de changer l'identité. Et je vais juste finir par quelque chose que j'ai vu souvent. Donc, ce paysage épigénétique, le dessin de Wellington, représente pour moi ce qu'on essaie de faire à la fac. Et donc, ces amphis qui sont un peu comme une montagne. L'éducation, c'est un système de reprogrammer, d'offrir à des jeunes et de plus en plus les moins jeunes la possibilité de recréer la potentiel de devenir d'autres choses. Et donc pour moi, c'est un peu un symbole de l'éducation. Et donc, j'espère que ce discours qu'on a commencé entre les disciplines, donc cette idée d'épigénétique comme un pont, comme un bridge, va nous aider tous de retrouver l'optimisme, donner cet optimisme à nos élèves, et peut-être trouver les moyens d'exploiter, de comprendre la plasticité sociale qui nous permettent de peut-être créer un monde meilleur.
Ah, quel optimiste. Si vous voulez en savoir plus sur l'épigénétique, il y a un 13 minutes de l'université. C'est des petits films faits par l'université où Jonathan explique l'épigénétique, qui est accessible à tout un chacun, et c'est en français si je me rappelle bien. Hein. Juste un peu dans le prolongement et avant de passer la parole, ce que vient de dire Jonathan, ça peut faire des échos pour nous. Il ne s'agit pas non plus d'en conclure trop vite. Mais que fait l'analyse? Elle part de fixations qui vont être bouleversées par un événement quelconque ou par une interprétation. Il va se créer un moment de désordre qui va créer une nouvelle plasticité du système qui immanquablement parviendra à la synthèse. C'est pour ça d'ailleurs qu'on n'a pas tellement à se préoccuper de la synthèse en analyse. Notre travail, c'est plutôt de créer un chaos supportable qui va être un moment de plasticité avant qu'on soit capable de recréer des trucs auxquels on risque d'être fixé aussi maladivement qu'avant. Le seul espoir, c'est qu'on soit fixé un peu moins maladivement à certaines solutions. Mais vous voyez, il y a des résonances là qui sont intéressantes. Alain ou Olivier d'abord?
Tout d'abord, merci à Eva Jablonka pour cette très brillante conférence, passionnante et très stimulante pour nous. Évidemment, François l'a dit très très justement. Au fond, moi quand j'ai fait de la biologie, on m'avait expliqué que Lamarck était quelqu'un qui était passé tout près de la grande découverte, mais finalement il s'était trompé et qu'il fallait l'oublier. Et du coup, l'attachement de Freud à Lamarck, je le rappelais à propos du Moïse, mais Freud vraiment était Lamarckien. Enfin, il n'a pas pris que la horde à Darwin, mais c'est le retour d'un certain lamarckisme dans le darwinisme régnant. Il y a là quelque chose pour nous de très intéressant. Ce qu'a dit d'ailleurs tout à l'heure Olivier, très justement, c'est aussi si pour nous l'ontogénèse récapitule la phylogénèse, alors on voit maintenant que d'une certaine façon la phylogénèse récapitule aussi des ontogénèses. Donc on a quelque chose là qui bouleverse un peu les partages freudiens, parce qu'il faut rappeler que Freud avait un espoir considérable dans la biologie. Chaque fois qu'il est embarrassé sur un plan théorique, il fait appel à la biologie, en espérant qu'un jour elle découvre cela, ce qui rendra totalement obsolète la psychanalyse, etc. Et c'était aussi le cas lorsqu'il distinguait l'accidentel et le dispositionnel. Il disait dans l'analyse, on peut agir sur l'accidentel, les accidents qui sont arrivés dans la vie de chacun, dans leur histoire, mais le dispositionnel, on n'a pas beaucoup d'effet là-dessus. On voit que là, d'une certaine façon, c'est une subversion freudienne radicale, puisque d'une certaine manière, l'accidentel a des conséquences sur le dispositionnel. Cette espèce de circuit est intéressant. Alors, la deuxième chose que je voudrais dire, parce qu'avec Catherine, avec Catherine Vanier, on avait, et Bernard Goltz, et Bruno Falissard, et Jean-Christophe Talabar, etc., et Arnold Munich, qui serait bienvenu ce soir, on avait mis en place un PHRC. Un PHRC, c'est un machin qu'on met en place dans les hôpitaux pour faire une recherche précisément sur le travail de Catherine, qui était notre protocole. Ce n'était pas une recherche comme celle qui a été faite sur les troubles du spectre autistique dont a parlé Eva Jablonka. C'était une recherche pour vérifier à travers un protocole incluant une étude de la méthylation. Et c'était tout au début. On a fait ça quand on venait de commencer à entendre parler de l'expérience avec les rats lécheuses et les rats abrutis. Et on avait eu le projet de faire une étude comme cela pour vérifier, d'une certaine façon, les effets d'un certain type de travail que peuvent faire les psychologues cliniciens ou les psychanalystes dans les services de réanimation néonatale avec les prématurissimes et les grands prématurés. Nous avions mis ce projet en place. Et je me rends compte qu'il y avait une erreur méthodologique. Alors Bruno Falissard était très enthousiaste, mais Jean-Christophe Talabar, qui est un autre méthodologiste, mathématicien, était plus réservé. C'est que nous pensions finalement que l'étude de la méthylation, et alors à l'époque on était dans les débuts, avec Arnold on avait une discussion où Arnold pensait qu'il fallait que le prélèvement ait lieu au plus près du cerveau. Donc ce qu'il avait demandé, c'est qu'on puisse faire un prélèvement à travers la fontanelle, et pour laquelle il aurait fallu demander l'autorisation des parents pour des bébés de 500 grammes, enfin un truc absolument abracadabrant. Mais je me rends compte que l'erreur que nous faisions après l'exposé d'Eva Jablonka, c'est que nous restions prisonniers d'idéologie du discours courant, qui était que nous accordions à l'épigénétique qui arrivait le même crédit absolu et totalitaire qu'à la génétique avant. Même si nous étions contre, alors qu'il est évident qu'il peut y avoir des modifications, il peut y avoir de véritables modifications des comportements et des effets sur l'enfant sans qu'il y ait pour autant une trace épigénétique répertoriée. Alors l'autre remarque que je voulais faire, c'est ce qui est assez formidable, et là il y aurait un travail à faire, puis certains ont déjà commencé, c'est qu'on retrouve dans ce que vous disiez sur les effets transgénérationnels, c'est une association d'idées, mais sur les effets de l'alcool, toutes les théories qui ont fleuri au cours du XIXe siècle en psychiatrie, de la dégénérescence de Morel, les roues macabres de Zola sont l'illustration, au délire chronique à évolution systématique de Magnan, 4 étapes suivant 4 générations, c'est l'illustration des parents boivent, des enfants trinquent, mais on voit comment, et cette idée, ces idées de ces psychiatres du XIXe siècle étaient des idées organicistes pour la plupart, c'était pas du tout. Donc il y aurait à revisiter avec l'épigénétique les intuitions assez géniales de ces psychiatres classiques qui, simplement par l'observation clinique, permettaient de penser à un certain nombre de choses. Effectivement, ils avaient la possibilité de botaniser parmi les humains comme jamais plus l'asile ou l'hôpital psychiatrique nous le permettra aujourd'hui. Enfin, un dernier point, j'ai une petite remarque, parce que j'ai trouvé que c'était une remarque pour ceux qui s'intéressent à Lacan. L'idée que le litérisme modifie le cerveau, ça m'a bien plu, on le sait. On sait que ceux qui ont affaire aux idéogrammes et ceux qui ont affaire à l'écriture alphabétique ne pensent pas pareil, et c'est vrai. Après tout, les lacaniens ici, ou ceux qui s'intéressent à Lacan, goûteront l'idée des effets dans le réel de la lettre, une question tout à fait importante. Et je voudrais rappeler d'ailleurs que les neurobiologistes qui travaillent avec les psychanalystes, je pense en particulier à Enregistrée et en Sermet, ont trouvé le point pivot de leur travail entre neurobiologie et psychanalyse autour de la notion de trace. Voilà. Donc, quand Eva Jablonka dit au fond, c'est quelque chose d'extrêmement riche pour les psychanalystes, quand Eva Jablonka dit que le discours, voire même l'effet symbolique, puisque elle a un livre sur l'évolution, il y a 4 dimensions, hein, voilà. Donc une qui est symbolique, je pense, et la manière dont elle en parle, on pourrait tout à fait, sans faire de grosses erreurs, l'appliquer à notre symbolique à nous, et bien qu'il y ait, à partir de là, des effets dans le réel, ça c'est quelque chose quand même formidable. Donc merci beaucoup, et je crois que c'est vrai qu'il faudrait qu'on puisse mettre en place, peut-être en rectifiant par exemple ce PHRC ou sur d'autres objets, les Rwandais vivant en France, il faudrait qu'on trouve un objet autour duquel on pourrait mener une recherche commune, car je crois qu'il n'y a là pas de contradiction. Pas de contradiction entre une méthodologie comme celle-là et puis ce que nous pouvons faire d'une façon absolument singulière qui est en tension contradictoire avec toutes les généralisations ou universalisations.
Alors, Olivier Putois, il y a Eva Veil qui est inscrite, Alexandre Moron aussi. Il y en a d'autres qui veulent la parole.
Moi, je ne vais pas monopoliser, parce que de toute façon, le contraste serait trop important, mais ça me paraît intéressant de rebondir sur le fait qu'au fond, et ça c'est une discussion qu'on a eue déjà, notamment avec Alain, mais c'est qu'au fond, comme quand Guilhem disait que pour la philosophie, toute bonne matière est une matière étrangère, bon, il ajoutait en disant que toute matière étrangère est bonne. Finalement, ce qui est intéressant, c'est qu'au fond, du point de vue de l'épistémologie psychanalytique, disons, ce type de raisonnement qui revient à développer de manière un petit peu méthodique des intuitions de sens commun sur les rapports entre les différents niveaux de causalité, au fond, ce type de raisonnement revient à essayer de... Enfin, ça offre la possibilité à l'épistémologie analytique, au fond, de voir ce qui, du point de vue d'une pensée biologique un peu modélisante, peut nourrir peut-être une phase nouvelle. Hein. Bon, c'est vrai que Lacan qui introduit la révolution structurale, au fond, pour lui, la science, c'était cohéré, grosso modo, donc c'était le galiléisme, etc. Bon, c'est quand même des choses extrêmement datées. Hein. Et là, il se passe quelque chose, peut-être, qui du côté de la biologie, peut venir un petit peu déborder, déborder ce qui est établi. Et peut-être, au fond, ces discussions qu'on a eues notamment avec Alex Moron, d'ailleurs, ce qui paraît intéressant, hein, c'est que, peut-être, en développant ce type d'investigation entre les différents niveaux de causalité, au fond, il faudrait se demander dans quelle mesure on peut pas utiliser ce type de modèle pour essayer de penser ces bridge principles, les principes d'articulation causale entre les différents niveaux de la réalité, qui sont au fond, c'est une manière épistémologique de formuler un petit peu le problème face auquel le Labex se retrouve. Et est-ce qu'on ne peut pas, c'est une question, on aura peut-être l'occasion de revenir là-dessus par la suite, mais est-ce qu'on ne peut pas, au fond, se demander si, en utilisant la médiation d'un objet complexe, par exemple sur le modèle de ce qu'Eva proposait là, mais il peut y en avoir d'autres, on ne peut pas essayer d'affiner cette question préliminaire que vous posez au Labex, de l'articulation des différents niveaux de réalité, en montrant comment ça fonctionne, en quelque sorte, à partir d'une étude de cas, pour en tirer des conclusions peut-être en direction de cette question que vous nous posiez. Voilà, je ne monopolise pas la parole plus longtemps, mais puisque vous alliez parler après, je vous envoie une petite ouverture.
Alors, Eva Veil, qui est psychanalyste et psychologue clinicienne ayant travaillé longtemps dans les procréations médicalement assistées.
Tiens, pardon.
Merci de ton apport. Après surtout les différentes discussions qu'on a eues ensemble. Et je trouve que ton apport ce soir nous permet de déplacer un peu l'antagonisme un peu idéologique entre l'acquis et le transmissible. La biologie de la culture, telle que tu nous la proposes, nous pousse à examiner plus finement tous ces différents segments explicatifs de nos disciplines. Et c'est peut-être la force, en tout cas qui m'a paru le plus intéressante ce soir. Souvent, nous, dans notre champ, nous nous référons à la plasticité psychique. Et pourtant, aussi vite énoncé, nous retombons dans nos causalismes particuliers et idiosyncrasiques. Comme le disait tout à l'heure Jonathan, quand tu parlais de la plasticité sociale, pour nous aussi, le psychanalyste, la plasticité psychique, de la plasticité psychique à la plasticité sociale, est une des difficultés de notre discipline. Quel chemin on peut tracer entre les deux, et pourtant il est important. Et pour reprendre la citation que j'ai beaucoup aimée, enfin c'est pas une citation, ou je sais pas comment tu as dit, il y a des chemins qu'il nous faut faire devenir des routes. C'est je ne sais pas si c'est chinois, japonais, mais peu importe. C'est assez universel. Et pour construire une route, il faut des acteurs différents et des matériaux hétérogènes. C'est peut-être ce que nous pouvons faire ensemble, l'hétérogénéité et le différent, mais pour construire une route, biologistes et praticiens des sciences humaines, et d'autres encore, et surtout autour de l'humain, qui est notre objet commun, même si c'est un peu ridicule de le dire sous cette forme, de l'objet humain et de ses possibles transformations traumatiques. Merci.
Alors, Alexandre Boron, qui est professeur de bioéthique, je crois, hein, c'est ça, à Lausanne, Genève, et qui est un des membres du conseil scientifique du Labex.
Oui, merci à Eva pour cette conférence qui nous fait partir dans beaucoup de directions qui mériteraient d'être réfléchies l'une après l'autre. Et comme l'a dit Olivier, je trouve qu'il y a une tâche assez minutieuse à faire, qui est d'identifier exactement les niveaux de réalité dont on parle, et je dirais même d'essayer d'identifier les malentendus à mesure qu'ils se forment. C'est un peu dans l'esprit d'explorer ces ponts, c'est important de voir ces malentendus. Dans ce que je vois se développer dans le domaine, je vois déjà plusieurs de ces malentendus en formation, et peut-être que j'en parlerai d'un, c'est que d'un côté, la recherche dont Jonathan à laquelle Jonathan a fait allusion, a une notion claire, relativement claire, des distinctions entre des effets génétiques et des effets épigénétiques. Et commence à avoir en tout cas des explications moléculaires pour certains d'entre eux, mais on est quand même dans le relativement clair. J'ai l'impression que ce qui se passe, c'est que maintenant, la relation entre génétique et épigénétique devient une métaphore pour un problème beaucoup plus flou, qui est les relations entre les gènes et l'environnement, ou entre l'inné et l'acquis. Mais ce n'est pas la même chose. Par exemple, les résultats que vous avez montrés sur la surdité et l'effet populationnel de l'apprentissage du langage des signes. Alors, je ne connais pas du tout ces recherches, mais le peu que j'ai vu de mon vieux background de généticien des populations, c'est que c'est de la génétique des populations classiques. Alors peut-être qu'en plus, il y a de l'épigénétique. Mais au départ, ça n'est pas évident que ce soit de l'épigénétique. Et donc, par rapport à ces différents exemples, je pense que c'est intéressant de chaque fois bien tracer la limite entre ceux qui relèvent d'une explication génétique classique et puis ceux qui relèvent d'un effet épigénétique. Le deuxième malentendu que je vois, c'est peut-être un malentendu qui vient de la référence à Wellington. Parce que Wellington s'intéresse à la biologie du développement et il découvre au fond, il développe des métaphores, la métaphore du paysage et de l'attracteur, qui sont en fait tirées des mathématiques, des systèmes dynamiques de son époque. Alors quand on reprend ces métaphores pour les appliquer à des phénomènes sociaux, réseau complexe d'interaction sociale, bien sûr que cette métaphore est pertinente, mais ce qu'il y a de commun, c'est simplement la métaphore, et éventuellement l'arrière-fond mathématique, qui entre-temps d'ailleurs a beaucoup changé. Mais ce n'est pas l'épigénétique. Il n'y a aucune nécessité à ce que cette métaphore du paysage telle qu'elle est utilisée par les sciences sociales aujourd'hui ait quoi que ce soit à voir avec l'épigénétique. Peut-être qu'elle l'a, mais ça c'est une question il faut commencer par la poser pour avoir éventuellement une réponse. Deux exemples du genre de clarification conceptuelle qui me semble être à l'ordre du jour.
Okay, I just want to answer a few of the things. I will start with, if I understood you correctly, you were talking about the kind of conflation that may happen between nature-nurture kind of thing, or heredity and environment, and epigenetics and genetics. I think that one of the interesting things is on this point is that the understanding of epigenetics makes the kind of dichotomy between the environment and heredity and genetics a little bit. It sort of dissolves a lot of the dichotomy between these two concepts, because you cannot have an epigenetic phenomenon, which the epigenetic phenomenon is by definition the product of an interaction of the genome with the environment. This is what makes, so it's a relational process, it's a relational concept, and therefore it is an integrative concept, which makes the dichotomy between heredity and development, for example, not very between genetics and development, and genetics and environment and environmental genetic effects and environmental effects, very, very much less clear. It's sort of by definition the epigenetic is the interaction. So if it isn't an interaction, it is not, we don't have to choose between environment and genetics. Right? This is the first thing, and I think this is why the thinking about epigenetic helps us in getting rid of classical kind of dichotomies in the thinking of geneticists and people from the genetic domain and from another domain. Now, I wasn't sure that I caught your point about population genetics. You were assuming that there is a strong genetic component. What is? Yeah, right. The conclusions can be correct. What I'm trying to say is not to get rid of genetics. Right? I'm a geneticist by training, so it's not. I'm not trying to get rid of genetics, but I'm trying to say that thinking in terms of classical genetics is obsolete today. Not because classical genetics, there's something wrong with it, but it is a certain level of description that is not sufficient for understanding the many phenomena that we're dealing with. For some phenomena, it's okay. If you want to understand genetic polymorphism between populations and so on, that's fine. It is informative for some things, but it is not informative for many questions. For example, it is not informative for a question of poverty. So we have to define the kind of problem that we have, and then we can find out whether or not the genetic analysis is relevant for this particular problem. Epigenetics is always dependent on genetics by definition, but it is dependent on genetic expression rather than on genetic differences. And that's all. So I think that this allows us to talk about gene expression, genetic expression, patterns of expression, and for many kinds of problems that are interesting and relevant for psychologists, psychoanalysts, and social scientists, this is of importance. Now, I agree with you that there is a tendency to think that epigenetics is going to solve all the world's problems. And in the same way that genetics couldn't solve all the world problems because it's at a particular level of description, epigenetics can do it. It's a kind of bandwagon on which a lot of people jump because it's very, very fashionable today. But it is a very useful, it's very useful that we understand it and that we will understand it more, and it will give us a lot of very useful information, just as genetics did. And it will be relevant for a lot of things, just as genetics is relevant for a lot of things. And for some things it won't. And, it is, you said that there is a kind of metaphorical use of epigenetics. And that we are using it. So I don't think that epigenetics that epigenetics is taking the place of environment. I don't think so. Because I think that epigenetics is, as I said, a relational process, a relational concept, whereas environment isn't. But I think that we have to be very, very careful about metaphors, and Wiener said that the price of metaphor is eternal vigilance. So it's true. We can't do without them, but we have to be very careful about them. Now, I'm not sure that I remember all the questions, and all the comments, but I think that, I understand that most of you here are psychoanalysts and you're interested in Freud and you're interested in his idea about the transmission of trauma and the role of collective trauma and its transmission. Now, I think that what we're learning from epigenetics is, at least for the time being, is that you can transmit some effects of trauma, but it is not content that you transmit, but rather a threshold of response. And this is a very different thing. I think, again, what I'm saying now is also one has to be very careful about it, because it is based mainly on work on mice and rats, and human beings are not mice and rats. And although, well, anyway, we're very different kind of species, and we're long-lived species and so on. And the symbolic aspect is enormously important in our species, very, very, and it has many, many very subtle effects also, which begin very, very early on. And I understand here that Kathleen, yes, she's working on children that were born very early, and she's drawing on work that was done by a group of people who were working on rats and who showed that it is very important for a young pup, which is born especially during the first 6 days of its life, in the case of a rat. For human beings, as far as I know, it's the first 6 weeks that are enormously important. This is a sensitive period, to be touched and to be physically touched. Physical touch is absolutely important for mammals, and it allows normal maturation of the nervous system. It has a lot of extremely important effects. Children, for example, in orphanages who are not touched develop a lot of terrible psychosis and terrible mental diseases as a result of that. And the work of rats has shown in a very convincing way that even within the normal range of variation in a population, the differences between a touching mother and a non-touching mother lead to long-term effects which are transgenerational, which are then reconstructed generation after generation. They are not transmitted through the gametes, but they lead to the reconstruction of development. And this is a very important effect, and there is an involvement of epigenetic mechanisms in this. And one has to make a distinction, and I think this is also a point you have made, that is very important, between epigenetic mechanisms and epigenetic inheritance, for example. Because there are epigenetic mechanisms all over the place. Always, in every developing organism, all the time. And they're very important. They're very important for learning, for example. But epigenetic inheritance is a specific, as I said, it's a component of epigenetic, and it takes place only under the, it's a specific kind of thing that we have to show, to prove, and to analyze. It is the same epigenetic mechanisms that are involved, but that's it.
Alors, Muriel est psychanalyste et professeure à l'université de Dijon.
Merci pour tous les intervenants et aussi ce, comment on pourrait dire, ce melting pot de culture, d'origine, et qui fait un mélange, une soupe originelle, à nouveau une nouvelle soupe, et je crois que cette soupe-là, elle prend et elle est onctueuse, délicieuse, et le fumé, c'est-à-dire l'odeur, nous offre de nouvelles perspectives où on a envie de goûter ensemble à tout ce que vous nous offrez. Alors peut-être, comme le temps passe vite, donc juste peut-être comme ça, une petite réflexion et une question un petit peu à tout le monde finalement, vous vous en emparerez si vous le souhaitez, c'était de dire finalement, je soupçonne, un soupçon, peut-être de faire parler la nature hein, Eva. C'est-à-dire que finalement, toute la nature, un peu comme les romantiques, hein, c'était une façon, vous avez parlé d'Ascala hein, tout à l'heure, mais le reprendre aussi dans le romantisme, où finalement la nature est un élément qui nous parle à notre âme de la profondeur de l'âme. Et je me disais peut-être que par rapport à l'ADN, donc le génome, je me disais peut-être que ce que vous souhaitiez, c'était non seulement de faire parler la nature et le génome, mais aussi de lui donner une temporalité, le temps qu'on pourrait percevoir à travers le génome, c'est-à-dire une sorte de narrativité, c'est-à-dire le génome comme une histoire interactive avec l'environnement, bien sûr sans cesse renouvelée. C'est-à-dire qu'en fait, on pourrait dire, comme Claude Bernard, de dire le milieu intérieur, nous avons le milieu intérieur, la mère originelle, vous l'écrirez comme vous voulez, mais on peut dire aussi que l'ADN, c'est aussi finalement notre façon de pouvoir réinterpréter le texte qui est là, sous forme de, évidemment, de protéines qui sont un peu fixées, hein. On connaît le langage de l'ADN qui ne bouge pas. C'est ça qui est intéressant, c'est que les lettres de l'ADN ne bougent pas, mais sans arrêt, l'ADN nous montre que c'est sans cesse en interaction ces lettres qui s'écrivent sans arrêt, et en s'écrivant, ces lettres transforment l'histoire. C'est-à-dire que l'histoire est transformée à partir de ces lettres qui sont immuables. Donc l'immuabilité de l'ADN, justement, nous permet d'inscrire une nouvelle histoire chaque fois. Voilà.
Je remercie Eva Jablonka pour son exposé si intéressant. Et en psychopathologie, on voit de plus en plus que certains diagnostics deviennent de plus en plus fréquents, comme les spectres autistiques. J'avais parlé, les troubles bipolaires, on entend beaucoup parler de ça. On parle d'un pourcentage de 5% dans la population générale. Et on peut penser qu'en psychiatrie, il y a eu toujours de modes. Il y avait des époques où on posait tel diagnostic et pas un autre diagnostic. Et est-ce que vous pensez que dans ce qu'on voit dans l'augmentation de fréquence de maladies mentales, est-ce qu'il pourrait y avoir une part épigénétique? C'est une question peut-être un peu difficile à répondre, mais quel est votre sentiment par rapport à ça? Est-ce qu'on pourrait penser que l'évolution, par exemple, de la culture peut faire que certaines maladies mentales peuvent devenir fréquentes, pas pour des raisons seulement sociales, mais aussi pour des raisons génétiques?
Well, I will briefly just, I don't know if I understood everything, because my French is rather weak. I mean, I think that I understand the gist of the question. So, if you are saying that it's a dynamic system that is being changed as it's being sort of read, I think if you're talking about epigenetics, it is a very dynamic system. The genome also is much more dynamic than we thought. The genome at the level of the DNA sequencing. And there is a recent book by James Shapiro that is documenting all the many changes that are happening to the genome itself as it's during development and evolution. So the epigenome, the kind of thing that I was talking about, certainly is a dynamic thing. And I think that in a way, what I want to say is we're giving this example of writing it's a metaphor that is very, very commonly used about DNA, that it's a text that is being read and interpreted and so on. And it's like a software that is being installed in different hardware. Well, I think it's a better way of thinking about DNA not as software but as data that is being fed into the developmental system. And in this way, it is not a blueprint. It's not the software is not in the DNA. The software, if you want, is in the cell, in the dynamics of the interpretive dynamics of the cell. This is the software, and DNA is one type of data that is going into this into the system. Now, this is a very difficult question. The question that you asked about about autism and other kind of mental diseases, if I understood correctly, is a very, very difficult question. There was a great hope with the Genome Project that the problems of autism will be solved once we have the genome. And they have not been solved. And that's why people have done this work, for example, looking at the identical twins to try to see the epigenetic aspects. Now, I know that different periods in history have been characterized by different mental illnesses. I mean, there was hysteria during in the period that Freud was working, which was quite common. There was there was I forgot how it was called, but in the United States in the 19th century, in the beginning of the century, there was a kind of mental tiredness, mental fatigue that was very, very common and was characterized. Neurasthenia, that's it. Neurasthenia that was characterizing William James and all, and many, many other people. But it was an epidemic, and we have this type of things. It's very, very difficult for me to say. With epigenetic, there is about autism, for example. There are many, many attempts to understand this stuff. We are also some people believe that it is that epigenetics have a very big input. It doesn't mean that genetics is unimportant, yes, because it doesn't mean that if you have an epigenetic effect on autism, there won't be a genetic effect on autism. I mean, they don't exclude each other. Yes, that's the first thing. But what is it? What is it about our society? Why do we have this autism epidemics? Why do we have so much big bipolar syndrome? It's very difficult to answer. One type of answer that people are exploring at the moment is that it has to do not so much with change, with societal changes. I mean, that's one type of answer. But with exposure to toxicity, to all kinds of medication that women were giving in the 1960s against to help with fertility and to help with pregnancy, to carry on pregnancy and things like that. And the idea is that maybe this kind of medications had an effect on the germline of the fetus. So they will be expressed not in the mother, but in the next generation, but in the grand offspring. This is one kind of possibility that is now being explored. It is a kind of toxicity that is cultural, yes, because it is due to a very, very strong medicalization of our culture, especially with respect to reproductive kind of aspects. There is another possibility that people are exploring, which is different, which is saying that people who are very strong systematizers, that is, that they have a very strong tendency to a certain type of personality, tend to marry in our society. There is a possibility of coming together of two systematizers, which was not which didn't happen before. Because, for example, women who were of this types would not necessarily meet people of this type because they were not going into computers, for example. But once you have this type of mating, the probability of an autistic child is increased because the gene combinations in the parents are such that they can. So there are many, many, many kinds of attempts to explain it. Some genetic, some epigenetic, and I really don't know. I don't know the answer of this. It is a very great enigma.
Bon, on va devoir interrompre cette soirée. Alors juste, ceux d'entre vous qui auraient envie de poursuivre cette discussion, mais là c'est plus avec des biologistes et autour des biologistes, il y a un workshop demain toute la journée avec entre autres Eva Jablonka dans la matinée et 3 autres collègues biologistes où on va travailler autour de ces questions d'épigénétique. Autre point, je tiens à remercier Olivier Putois qui a été le cicérone de Eva depuis qu'elle est arrivée et qui a grandement contribué à l'organisation de cette soirée. Et enfin, et surtout, merci à Eva, parce que j'espère que cette conférence et les promesses qu'elle contient, nous allons être capables de les tenir. Donc merci vraiment à toi.